So yes, I'm going to speak to you very briefly about what we know about the genetics of scoliosis. Um, and let's see, my advance is not working. Okay, here we go.
There you go.
Yeah, okay. So you've heard a great introduction by my predecessor about scoliosis. This audience knows scoliosis very well. It's the poster child for orthopedics, and it is a spinal curvature that can develop at different times in life. And so in genetics, you heard about scoliosis being divided, described by different sort of clinical presentations. And in genetics, oftentimes we describe it by the age at onset. And so there's a form of scoliosis called early, and congenital that you heard about earlier is an early onset form of scoliosis. So the early onset group is really many, many, many different diagnoses, as you see here by all these wonderful pictures that were provided to me by my colleague, Dr. Ramo. And then there's another form of scoliosis that you may have heard about that people call adult scoliosis. And this tends to be secondary to some of the things that happen during aging. For example, disc degeneration and osteoporosis. And then, of course, the most common form, which you've heard about previously, is the adolescent onset form that happens in otherwise apparently healthy children during the adolescent growth spurt. And so what do we know about these from the viewpoint of genetics? And when we talk about genetics, we're talking about the area of biology that's the study of inheritance. And how are these disorders inherited? How do the genes interplay? What is the DNA variation and the interactions, and how do they interact with environmental factors to cause disease? So that's sort of the global definition of genetics. And that's what some of us spend a lot of our time working on. And so in my world, in genetics, we think about genetic disorders in sort of two big buckets. And one is what we call single gene disorders. And this is kind of what we tend to think of when we talk about a genetic disease. And that's where, as you see on the left, this is a pedigree or family tree, and the squares are males and the circles are females, and black symbols represent someone who has a particular disease. And you can appreciate very quickly looking at this family tree that this disease is inherited in every single generation. It's what we call a dominant disorder. And so we know in that situation, this is likely caused by a single gene. One misspelling in the genome causes this disorder to arise in a family. And these days, with modern technologies, those are pretty easy to test and identify in patients. And then there might be another type of single gene disorder that's actually misspellings inherited from both parents and arising in a child, but it's still considered a single gene disorder that's causing that. Many of the early onset forms of scoliosis are single gene disorders. And again, remember I told you that early onset is sort of a heterogeneous mix of many different diagnoses. And many of these are genetic. And so what we're finding more and more is that sometimes these patients that present with scoliosis early in life have an unrecognized genetic disorder. And we encourage our colleagues to send them over for clinical genetic testing. But I'm not going to talk about adult scoliosis. That's really more of an environmental and aging thing. But now let's talk about adolescent idiopathic scoliosis, or AIS. This is the big one, and this is the one that probably most of us in the room are thinking about. Again, the most common form of scoliosis. And number one thing take home here is that adolescent idiopathic scoliosis is what we call a complex genetic disease. It is not a single gene disorder. It is not a single misspelling like you saw in my previous slides. This is a situation where many genetic changes in each individual patient come together and increase the risk of developing AIS. And that's why we show it, as you see on the left, it's as if you have many puzzle pieces that come together and increase the risk of of this disorder. And so over the years, we have identified many of those puzzle pieces, and I'm just showing you clips of some of the papers that have been published. And so what has this taught us? What it's taught us is that, yes, indeed, it is a very complex disease. There are more genes to be discovered. But in studying what these genes do, we have learned in the past few years that the cartilage in spine is a relevant disease tissue. Many of these puzzle pieces, actually, these genes function in the cartilage of the spine, in the intervertebral disc, and in the growth plates near the IVD. And so now research is very focused on how those players are coming together to cause AIS. And there's more research to be done to find more of those puzzle pieces. So what about genetic testing for AIS? That's something that's very important to us, and obviously it's very important to patients. Is there a test to tell a patient if they're going to develop AIS? And the answer is not yet, but it is coming. So I'm showing you here a picture of my friend and colleague, Dr. Nao Otomo. He's a researcher in Tokyo, Japan right now. Currently, he's doing a fellowship in my lab. And he has published a paper showing that if you test for many of those puzzle pieces, if you will, a selected panel of genes, it's called a polygenic risk score, you can sort of divide patient groups into high risk versus low group. So in his test, by using these selected genetic variants, the test showed over 60% sensitivity and specificity for distinguishing the high-risk groups from the low-risk groups. And if they put other information into their algorithm, for example, body mass index, that improved the sensitivity and specificity of the test. So what is all that? Well, and what about testing for AIS progression? In his study, he also showed that these factors could distinguish high Cobb angle patients from the lowest Cobb angle patients. So what does all that mean? The test is not good enough yet for clinical use. We want it to be much higher sensitivity and specificity, but it is a very good start. And so with more research, we are very focused on improving that test, number one, and number two, developing a test that would be useful for many populations around the world. So to summarize, we've come a long way in understanding AIS, and I show the slide on the right because obviously this is a very old picture. AIS has been around for a long time, and with modern technologies in the last 10 plus years, we're really starting to get a handle on what is causal and factors that could help us with better diagnosis and treatment in the clinics. So as I was saying, research is continuing to understand not only genetics, but in this case, genetics, it's an interplay between genetics, between sex. You heard earlier that females are at much greater risk for progressive disease than males. And so there are hormonal influences and obviously the mechanical stresses of growth. So again, with a major goal of leading to better testing and prevention of this disorder. So I thank you very much. I thank you so much for your attention. I always thank the patients and families who are the absolute drivers of the work that we do, and all of my wonderful collaborators.